So I hope my voice is clear and you guys are getting my voice. Chalo, fine. So we will start the session now. So hey guys, welcome you again. Now we are going to talk a few very funny things today. In fun, we will understand the major concept of biotechnology. We can say the principles and process, which is like grade 12 chapter. And this biotechnology is very important for your board examination as well as for your NEET examination. From NEET point of view, this chapter is like high scoring chapter, very small chapter, but it's a high scoring chapter. Now in this chapter, if you talk about NCRT in NCRT book, it's given like khichdi, it's complete mixture. So to understand that NCRT, we need to read NCRT in a systematic way. Okay. So that system, I'm going to explain you now how you can study biotechnology and how we can start, in which way you have to go through. Now, this entire topic will be based on the daily life what we do, and I'm going to explain this concept how we cook, how we use the kitchen. That's why I give this heading: recipe of producing biological products using biotechnology. So first let's understand what is biotechnology. Okay, the heading biotechnology. So you by this term biotechnology, you can understand that bio means biological, biology, that is your, we can say, life, and technology, you can say technology means equipments, or we can say some tools we use, whatever tools we use, all that tools, and then which we use with the help of this biology, that is your microbes, we can say microbes, or other host like yeast, etc. So if we use these life, and if we use the technical things, like for example equipments of molecular equipments, and if we use this molecular equipments on this biology, that is microbe or life, we can produce some product which is very much beneficial for the human mankind, or we can say mankind, or human life, okay, or animal life, okay. So it is very much beneficial. So that's what we are going to study. Okay, biotechnology. Now in this we will study the principles. Principles means what is the principle of biotechnology, and what are the different processes. Processes means steps involved in order to get our final product. Okay, product can be anything. It can be hormones, or it can be enzymes, anything, any product. Okay, any product. Now let's understand how we can produce product. Okay, what can be the product, why we need to produce any product from any biotechnology, or any why spending so much of money on this, and we need insulin, or we need enzyme, why we need it. For example, let's say that is a person A. Okay, that is a person A who is having good amount of insulin production in his or her body. Okay. Now a person B, this person is not having enough insulin. Okay. He is not having enough insulin, which means he is deficient. So what will happen to this person? This person will suffer from diabetes. But this person will remain healthy. Okay, this person will remain healthy. Now, how we can treat this person? How we can improve this person's life? How we can treat diabetes? So by giving the person passively insulin, है ना? By giving insulin passively. Now, in order to give this person insulin passively, we need to produce this insulin, right? We need to manufacture this insulin. But from where we manufacture this insulin? Insulin manufacturing. So insulin, as we know, insulin is a protein. Insulin is a protein produced by, insulin is a protein produced by beta cells. Insulin is a protein produced by beta cells of islets of Langerhans. Islets of Langerhans, which is present in pancreas. Which is present in the pancreas. Now, this pancreas, they have islets of Langerhans, and this islets of Langerhans will have beta cells. For example, if we say this is your pancreas, in this pancreas there will be Wilson's duct, and this is ampulla. Here there will be like cystic duct, and in between this cystic duct there will be so many ball-like structure, and these ball-like structures, they are called islets of Langerhans. Now, if we magnify this ball, it will be like this. So, for example, there will be alpha cells, there will be beta cells, there will be gamma cells, there will be delta cells here. All these cells will be present inside the islets of Langerhans. In this, alpha cells will secrete glucagon, beta cells will secrete insulin. Now, what we need this beta cell. So, let's say this is a beta cell. Okay, this is a beta cell. Now, this beta cell, they will have a nucleus, they will have genetic material, and in this genetic material there will be a small amount of gene. Let's say this is the genetic material of this beta cell. In this, maybe from here to here, this gene, they are responsible for the production of what? Insulin. Okay, which is a protein. So this gene will be transcribed, then it will be translated, and then insulin will be produced. Okay. So let's say here we have a person A who is good, or we can say he is healthy. Okay. This person is healthy. We'll use the term healthy. Okay. This person is healthy. Okay. Which means he is having good amount of insulin production, which means this person's gene which is present in the beta cells, they are working right, so they are producing enough amount of insulin. Let's say there is a person B. Take a person B. This person is not having insulin, so we need to transfer this insulin to this person and inject this insulin into the B's body, and after that he will also be healthy, or we can say his diabetes we can reduce, right? So how we can produce this insulin? We need to produce the insulin. So we need to isolate the gene which is responsible for the production of insulin. Then we need to transcribe this artificially. We need to produce mRNA artificially. Then we need to produce what? We need to produce protein artificially. And what will be that protein? That protein will be your insulin. Okay, that protein will be your insulin. So all these steps we need to perform artificially. We need to perform, and if we perform this artificially, for this taking the gene from the, for example, let's say we have 30,000 genes. How many genes we have? We have approximately 30,000 genes in every cell, and these genes, they are responsible for so many functions. In that, one function is your insulin secretion. So we need to take this one small part of this gene which secretes what? Insulin. How precise we have to be. So in order to take this gene out, then we need to transfer this gene somewhere so that the organism can transcribe or translate. So we need to have that much precision, whatever it is. Okay, you have to be very precise in order to produce the gene, or we need to produce or isolate the gene, and in order to produce the desired product, which is insulin. So similar to this, we can produce enzymes also. we can produce so many other components. Okay, we can produce enzymes, we can produce hormones, we can produce proteins, we can produce like so many different products. So how we can do that? For this entire process, we need some good amount of technology. Okay, some good technology. That technology is your biotechnology. Okay, that technology we use in the field of biotechnology. Okay, what is that technology? We need to first, acha, chalo. How we cook? We used to cook, okay, in our kitchen. We used to cook what in our kitchen? First, we need to chop the vegetables, right? First, we need to chop the vegetables. In order to chop the vegetables, okay, what we use? We use scissor. Huh? No, but some people they use scissor also in order to chop like beans, beans or small vegetables. Some people they use scissor, but it is not the right tool to use. So what is the right tool to use? The right tool to use is knife. Okay, knife. We need to chop. For example, if this is the lady's finger, actually the lady's finger will be green in color, but still fine, no problem. Let's say this is the lady's finger. Okay, now yeah, it is looking like lady's finger. Nice. Chalo. If this is a lady's finger, if this is the lady's finger, so we need to chop this. First we'll cut this portion, then small we'll chop. Okay, we'll chop into small pieces. Okay, we'll chop into small pieces, right? Okay, then after cutting this into small pieces, okay, for example, this is one small piece, this is one small piece, this one small piece, then we will fry. So what we use? Knife to cut this entire portion, right? Similarly, if you want to cut this lady's finger, we need knife. Similarly, if you want to cut the DNA, take care. If you want to cut the DNA into pieces, we need scissor. Not knife, scissor. Okay, this scissor we are using to cut DNA, which is a life, right? Bio. So that is why this is called. So this is called molecular scissor. Okay. All right. So yeah, fine. Chalo, fine. Okay. So this is called molecular scissor. Now, using this molecular scissor, what we can do? We can cut this DNA into fragments, right? So first thing what we will do? We will cut the DNA into fragment. But before cutting the DNA into fragment, we need to isolate the DNA. Okay, we need to isolate the DNA. So that will be the first thing. In order to cut the lady's finger, we need to bring the lady's finger from the field or from the market. So first thing what we can do is, to isolate the DNA. Okay, first is your isolation. First step is your isolation. So first we will isolate the DNA. From where we will isolate? That's a question. From where we will isolate the DNA? We will isolate the DNA from a healthy person. Okay, we will isolate the DNA where? From a healthy person. So let's say healthy person is Mr. A. Mr. A is very healthy. So he is having gene. So this is the DNA. So we will take the gene of Mr. Healthy person. Now this gene we will take out. Okay. Now what we will do? We need to cut a small piece of gene from this entire gene, right? So first step is your isolation of DNA. This is the first step we need to do in order to cook the product. Okay, in order to get some beautiful product. So first thing is isolation of DNA. After that, what we do? We will chop, right? Second thing is chopping. What we'll do? The second thing will be your chopping. Chopping. So here, instead of chopping, we use the term fragmentation. We use the term what? Fragmentation. fragmentation. So we will cut this DNA into multiple fragments. What we use during for this process? We use molecular scissor. Molecular scissor for this. Okay, we use what? Molecular scissor. I will explain what is molecular scissor, what are the different types of molecular scissor, like in some other video. Here we are just understanding the steps. Okay, recipe only. So second thing is fragmentation of this DNA using molecular scissor, and the name let me write, they are called restriction enzymes. Okay, restriction enzymes. So then we will chop. After fragmentation, we need to select, for example, after this process, what we will get? We will get a DNA fragments like this, but it may be in it may be in groups and clusters, fragment clusters we will get. From this, which one is our insulin producing gene? How we will come to know? Then we need to first we need to separate these DNA, these genes. Okay. What we need to do? We need to separate this. But even then, we will not understand which one is our gene. Okay, it will not be visible clearly. So what we will do? Okay, third step is your separation. Third step is your separation of DNA fragments. For this process we use what? We use agarose gel electrophoresis. ठीक है, agarose gel electrophoresis we use in order to separate the DNA fragments. Okay, we got the DNA fragment, but this DNA fragment, we got this DNA fragment, but this DNA fragment, it will not directly, or for example, if we take the small amount of DNA fragment and if we place it on this container, okay, or let's say this is a plate, if we place this DNA fragment on this plate and if we leave it as it is for 10 days, 20 days, 30 days, do you think this DNA fragment will produce insulin? No. We need to transcribe this, right? How we will transcribe? We will place it on the plate and we will leave it as it is. Do you think it will transcribe? It's like they are self-transcribing structures? No, they don't do. So we need to transfer this small fragment. This small fragment we call it as gene of interest. What we call? Gene of interest or foreign gene. Foreign DNA or foreign gene. Okay. Now we need to transfer this foreign gene or foreign DNA to a structure which can replicate, okay, which can produce product, which are self-replicating. Okay. So what we can do? For example, let's say there is a person who is like, chalo, you guys are, one of you guys, they are like working in a MNC, or let's say you are owning one huge MNC company. Okay, you are the CEO and you need to sign so many documents and you have one beautiful secretary to do that also. Okay, to present you the paper and you will sign the papers. And now your secretary, she is very, like, what we can say, greedy, and she wants to own your company. So what she will do? In a bundle of papers, so she is giving you so many bundle of papers. Okay, so many bundle of papers to sign, and in that bundle she is attaching one more paper, one more document, in which there is a document, it is a document to change the property, I mean attorney, or we can say power of attorney, change the ownership, we can say in simple terms, change the ownership. And you trust her, and you will first check the first paper, second paper maybe, and then you will put sign everywhere, and ultimately you will put sign on this sheet also. On this sheet also, unknowingly you will do that. How you will do that? Unknowingly. Okay, because you are signing this sheet also. Maybe evening she will call and she will tell you, from tomorrow you no need to come to the office because now I am the owner of this office. Okay. Anyways, so what she did, she fooled the owner, right? She fooled the CEO. Similarly, we need to fool the CEO. Okay, we need to fool some organism. The organism whose natural function is to transcribe its own DNA. So we will add our DNA into their DNA, and so they will just think that it's my own DNA, and it will transcribe it. Okay, and we will get our product. So we need to fool that. So we need to find out who is that fool. We need to find out the fool. Okay, we need to find out the fool. How we'll find out the fool? So we have some criteria. We will discuss that in another lecture. First thing, who is that fool? So that fool is, we can say, we should not call it as fool. They are giving product and we are calling it fool, but still we are going to fool it. That's it. We are going to fool the organism, and that will be called as vector. That will be called as what? Vector. Okay. So we are going to transfer the foreign gene or foreign DNA. We are going to transfer the foreign gene or foreign DNA into a vector. Okay. And what can be the, I mean, what is a vector? Vector is a transporting vehicle. Vector is a transporting vehicle. And who will be acting as a vector? Any bacteria, fungus, or any plant cell, or any animal cell. Okay, and other things. We have so many other vectors. So now these can act as a vector, which means we are going to fool the bacteria and we are going to transform the gene of interest or foreign DNA into the bacteria, so that the bacteria can transcribe their DNA and we can produce our product. Okay, we can extract our product. So the fourth step is we are going to isolate the DNA of bacteria so that we can transform. So isolation of vector DNA. Vector DNA is the next step. Isolation of vector DNA. Then fifth one. So for example, if this is a vector DNA, it's already complete circle, and we need to add extra gene. We need to add, so we need to remove some small segment of the gene, and then we can add our gene of interest here in this region, right? So we are going to use the same enzyme here. So that enzyme will cut the small fragment of DNA and it will remove the small fragment from this area. Let me draw like that. Okay, for example, if this is small part, so small part is removed from this area so that we can transform our gene of interest. So let's say this is the gene of interest. Okay. So after isolation of vector, we need to cut the vector. Okay, cut the vector. Then what we will do? We will transform, or we will transfer the gene of interest on vector DNA. And now after this process, this entire DNA will be called as, okay, now we will call this entire DNA as recombinant DNA. What is this DNA called? Small r DNA. Okay. So which means we are producing recombinant DNA first. So these are the few steps to produce what? Recombinant DNA. Now we have a recombinant DNA. And what is the process called? Recombination. Recombination. What is the process? Recombination. What is recombination? The process of producing recombinants. Okay, recombinants, which means this is a recombinant DNA. Clear? So this is the step of producing recombinants or recombination, but this is not the step to produce product. This is the few step. Okay, this is like first half of the process. Next, what will happen? So after producing recombinant, let's say we already have what? We already produced recombinant. So now this recombinant, we need to transfer this recombinant into host cell, because if we place this again, this DNA into any container, if we place the DNA here, it will not self-replicate until unless we transfer this plasmid DNA into the organism. So this organism in which we are going to transfer this rDNA is called host. So transfer of rDNA into host. But before transferring, we need to prepare the host, we need to make host competent, and there are so many other concepts in between. We will discuss that in a separate video. This process of transferring our DNA into host, it also requires some steps. Okay? But we will study again that in a separate video. What is the process called? The process called transformation. Transformation. What is that process called? The process called transformation. Transferring the rDNA into vector, any of the vector. Okay, we can transfer. That is called transformation. So next step is transformation. So transfer of DNA into host. What is the next step? Seventh step. Culturing or growing the host so that we can get multiple copies. Okay, the next step is culturing. After culturing, what will be the next step? What will be the eighth step after culturing? After culturing, we need to extract the product formed. Then what we need to do? Purification and packaging. Last but not least, quality testing and distribution. So this is the 10 basic steps involved in producing the products and packaging and distributing it. Okay, 10 steps, basic 10 steps. In between there are so many steps we have missed, right? The process we have missed, so that we will discuss in a separate lectures. Okay, these are the steps. Now in a concise, in a brief way, we can have like 6 steps we can say in order to produce recombinant DNA. As we already saw, recombination, or we can say rDNA, production of rDNA. What is that? First one is isolation of DNA. What is that first step? First step is your isolation of DNA. We need to isolate the DNA. Then second step, we need to fragment using restriction endonuclease, that is restriction enzyme. What is the third step? So this is the first, second, third step. Third step is isolation of desired DNA fragment. Okay? Using gel electrophoresis and other things. Then what is the next step? We have seen ligation of the DNA fragment into the vector. Transfer the vector to produce recombinant DNA. Okay? Then transferring the recombinant host DNA into the host cell. That is your transformation. After the transformation, we need to culture the host cell at large scale. Okay? For this we use bioreactors. Okay? We use what? We use bioreactors for this. Right? And for ligation, we can directly use sticky ends, or let's say we use DNA ligase for that process. Okay? These are called molecular gum or molecular glue. Okay? So these are the basic steps. After the extraction of desired protein, we will purify, package, and will send later. So these are the basic steps involved in order to produce the product. Okay? In order to produce the desired product from the gene of interest. Okay? Using biotechnology. I hope this entire step was clear. Okay? And it will be clear to you guys, and it will be beneficial also. Now few things, like we need to understand few basic things. What is the difference between recombinant and non-recombinant? Second, what is the difference between transformant and non-transformant? Number one, recombinant. Let's say this is a plasmid DNA. This is a plasmid DNA from the host DNA. Okay? And then this is your gene of interest. Okay? We need to transfer this in order to produce. We need to transfer this in order to produce what? In order to produce, this is your gene of interest. Okay? Foreign gene. In order to produce the combination. Now from this normal vector, it became what? Recombinant. So this will be called as recombinant. Now which one is non-recombinant? For example, the plasmid which is not having our gene of interest, they are considered as non-recombinant. Okay. Then what is transformant? Now this gene of interest, or we can say this rDNA, okay, this rDNA, we need to transfer this rDNA into host. So let's say here we have host, okay, and now in this host we have transferred our recombinant DNA, which means this host, they are already carrying our recombinant DNA, and they are carrying their own DNA, genetic material also. Anyways, they are carrying our recombinant DNA or recombinant DNA. Yes. Now this particular bacteria who is carrying a recombinant DNA, they are called transformant. And let's say we have one more bacteria, okay, some groups of bacteria in which they do not have recombinant DNA, they have generally non-transformants. So these are considered as they are carrying non-recombinant. So these are considered as non-transformant. Okay? Recombinant means the vector which is carrying a gene of interest, they are called recombinant. Non-recombinant, the vector which is not carrying any gene of interest, they are called non-recombinant. Transformant, the bacteria who is carrying recombinant DNA, they are called transformant. And non-transformant, the bacteria or the host, any host, it can be bacteria, yeast, any host which is not carrying the transformant, or which is not carrying the recombinant, they are called non-transformant. I hope I am clear to everyone. Okay? So these are the differences. Okay? Sometimes these will confuse you people, so please be aware of all these things. Okay? So remaining the processes, the steps and the different procedures like gel electrophoresis, how we can use molecular scissors, each and every procedure we will come up with a separate videos. Okay? And then in between I am also doing the question answer discussion also, that will also come in between. Okay? So I'm trying to give you something always, like some content, so that something can be beneficial for you people. See, listen, I don't have enough time, so because of that I'm not getting enough time to prepare videos, but still I'm trying to do my best. Thank you so much for listening, and I hope this will be beneficial for you, and next we will come up with some special video like this in a few days. Thank you so much for joining. Have a great night. Thank you.